Hi there. My name is Lauren. I'm a second year graduate student in the Developmental and Stem Cell Biology program at UCSF. I'm originally from Chicago and did my undergraduate studies at the University of Illinois Urbana-Champaign. And it's here that I found my passion for research while working in Dr. Bruce Fowkes' lab. I then took some time off and worked as a lab tech at the University of Colorado in Dr. Linda Barlow's lab, which seeks to understand the mechanisms behind taste receptor development and regeneration. And it was during this experience that I became passionate about stem cell biology and saw the power and potential that this research had regarding potential personalized medicine treatments. And now, as a second-year student, I've joined Dr. Jeroen Roos's lab, where I plan to study the regenerative response of the small intestine. Intestinal epithelium is constantly renewing throughout adult life from stem cells that reside at the base of the crypt. These cells are self-renewing and differentiate directly or indirectly into all cells within the intestine. Remarkably, ablation of stem cells in mice upon injury doesn't disrupt homeostasis. Instead, recent evidence in the field supports a model where various cells that line the intestine can dedifferentiate to replenish stem cells. However, which cell types are responsible for this replenishment remains controversial, and the mechanisms that regulate this process are still unclear. Stem cell fate decisions are dependent on tight regulation of protein synthesis. mTOR is a critical coordinator of protein synthesis, and mTOR signals are regulated by RAS-GRP1. Specifically, RAS-GRP1 and mTOR signals have been implicated to drive translational activity, thereby promoting immune cell fitness. Intriguingly, deletion of RAS-GRP1 in mice during intestinal injury decreases mouse survival rates, while mice that express RAS-GRP1 survive. I therefore hypothesize that RAS-GRP1 promotes the fitness of the intestinal epithelium during injury by driving cellular plasticity via mTOR and protein synthesis. To test my hypothesis, I first aimed to use mouse genetics and multiple injury models to characterize RAS-GRP1 as a cell-intrinsic fitness signal that promotes plasticity in the epithelium of the small intestine. Next, using similar mouse models, organoid technology, and assays used to understand protein synthesis, I aim to elucidate the mechanism of RAS-GRP1 and mTOR signaling in the small intestine during injury. Finally, I'd like to thank the ARC Foundation for awarding me this fellowship and providing me with the opportunity to share my work.